All right, so pregnancy, right? Fertilization until birth. We use some different terms here. Embryo and fetus. Sometimes we use those terms interchangeably, but they're really not interchangeable. The embryo is up until week 8, and the fetus week 9 through birth. And we'll talk about why we have that distinction between those periods in just a second. So there's a rapid growth thing that happens here between 8 and 12 weeks. Like 8 and 12 weeks, it's gonna more than triple in size. But 8 weeks still an embryo, 9 weeks fetus. The oocyte after ovulation is viable for 12 to 24 hours, and sperm cell viable 24 to 48 hours. So you have a window for fertilization. Hypothetically, for fertilization to occur, the sperm has to meet the egg somewhere in this time period. So sex has to happen like 2 days before or a day after ovulation. That is a much broader window than you would expect it to be. Like, it's not just a 3-day period. Now that's the optimal period for fertilization, but fertilization can totally take place outside of that window. Like 1,000% it happens. 6 children. 1,000% this happens outside of that window. So fertilization, when, man, my youngest is 2, and the one before that is 9. Outside of the window. So fertilization, when the sperm meets the egg. The attrition rate for sperm cells is really high here. Not all of them are going to make it. There's 100 million of them, so I mean, they're expendable. The female reproductive system is going to kill a lot of them. Some of them are going to make it through the cervix. Some of them are going to go the wrong direction because you've got 2 uterine tubes, only 1 oocyte hypothetically from one side, so some of them are going the wrong direction. Only a few of them actually reach the uterine tube. So somewhere between 100 and maybe a few thousand on a good day might actually make it there. In order to actually get there, right, they have to move, and that happens in the male reproductive tract. We talked about that last time, right? They mix with the secretions from like the seminal vesicle, and they start moving. But in order to actually fertilize the oocyte, they have to go through a process called capacitation. The membrane has to be weakened by the secretions from the female reproductive tract. And when that happens, it exposes that acrosomal cap on the head of the sperm cell. That acrosomal cap has enzymes that will digest little tiny holes and sort of the outer coating, the zona pellucida around the oocyte. In order to fertilize the oocyte, it's got to get through this. And there's going to be hundreds of them sort of clustered around the egg cell undergoing this reaction. These enzymes are going to be digesting a hole on the outside. Only one of them is going to actually fertilize that oocyte. So it doesn't matter like the first one there, the second one there, the order in which they get there is not super important. It's which one of them makes it through that outer layer with these enzymes. When those enzymes accomplish a goal and they break down that outer layer, the head of the sperm is going to approach the oocyte, and that acrosomal process binds to receptors on the oocyte. And when that happens, their membranes fuse. As soon as that happens, we're going to see a reaction happen in the oocyte that prevents other sperm from binding. Only 1 sperm cell will be allowed to fertilize the oocyte, because we don't need extra chromosomes. So, right, 23 chromosomes in that sperm cell. And in just a second, you'll have 23 here. Right now you don't, but in just a second you will. Remember that the oocyte doesn't undergo meiosis II until fertilization. So as soon as that sperm cell binds there, and the nucleus comes in, the oocyte ditches those extra 23 chromosomes. We get 23 and 23. If we had more than 1 sperm cell there, we'd have too many chromosomes, and that's not going to produce an offspring. So we have to block what we call polyspermy. Polyspermy would be multiple sperm cells fertilizing the oocyte. So we have to block that. As soon as that the sperm cell enters the egg, we get a calcium surge from the endoplasmic reticulum in the oocyte, and it causes what we call a cortical reaction. The cortical reaction releases enzyme that destroys all the other receptors. So as soon as that first sperm cell binds, all the other receptors are dissolved. So now there's not a place for any other sperm to bind. Not only that, that calcium is going to pull water into the oocyte, and it's going to sort of swell up, and any sort of sperm that are hanging around get pushed away. So we have a fast and a slow block here. And here we can see that. So now here's our sperm cell binding to that receptor, and then here's our cortical reaction, and all those receptors are being dissolved in the membrane. So you don't get more than one sperm fertilizing the oocyte. As soon as that happens, we undergo meiosis II. Like I said, meiosis II doesn't happen until fertilization. So the calcium surge that causes that cortical reaction also triggers meiosis II. So we shed those extra 23 chromosomes. In the meantime, the nucleus starts to swell up, the sperm nucleus starts to swell up. They're going to come together, and we've got 23 and 23, and the chromosomes recombine, and now we have a fertilized oocyte. A fertilized oocyte is called a zygote. So here we see that. So there's the sperm nucleus, it's really tiny. Here's us shedding those extra 23 chromosomes. Sperm nucleus, oocyte nucleus, they come together. You see the little mitotic spindles forming there. And now we've got a new nucleus, new cell, right? 46 chromosomes. It's going to start dividing. In meiosis, we're splitting the number of chromosomes in half. Right? We're going from a diploid cell that has 46 chromosomes to a haploid cell, which has 23 chromosomes. When the chromosomes don't separate evenly, it's called non-disjunction. And if they don't separate evenly, that means that the cells, the daughter cells that are produced here, might have one too many chromosomes, or they might not have enough chromosomes. We might get one with 24 or one that has 22. Any abnormal number of chromosomes is called an aneuploidy. Now, most aneuploidies are not going to produce a viable embryo. They may develop partially. There may be severe deficits because of chromosomal abnormalities. There are a few of them that we consider quote unquote compatible with life, but there's not a lot of them. So most of these aneuploidies, it either doesn't result in a viable pregnancy, or the embryo is lost very early on in development. Some of them, even if they do proceed all the way through the pregnancy, there's not a very high survival rate after birth. So we'll talk about some of these aneuploidies. First, here's non-disjunction. So remember what's happening in meiosis. So we start with that stem cell, make copies of the chromosomes, split them in half, split them in half again. So what should happen here is, right, each one of these has one of those little squiggly lines. Here we have non-disjunction, and now these are short chromosomes and these have extra chromosomes. This is non-disjunction in meiosis 1. Here's non-disjunction in meiosis 2. This is typically what we'd think of. So these have the normal number. That one's down one, and this one's got one too many. If you have only one of the pair, it's called monosomy. If you have more than one, if you have more than two, so if you have three, it's called a trisomy. The most, I suppose, survivable of the aneuploidies are aneuploidies of the sex chromosomes. So if you'll recall, we have all these chromosomes, 23 pairs. The last pair, we call it the last pair, is they're the sex chromosomes. So those are your X and your Y chromosomes. Female, male. Here's the thing, that X chromosome is necessary to be alive. Without an X chromosome, that is not an embryo. The Y chromosome, all that Y chromosome does is make the embryo male. That's it. So let's look at some non-disjunctions here, some aneuploidies. This is Klinefelter syndrome. Klinefelter syndrome, 47 chromosomes, XXY. So what happens here is that on the oocyte side, we get non-disjunction. So when what should happen, right, is you have that cell, and I'm going to simplify things here, and we're going to just make it one process instead of the two, like meiosis 1 and 2, right? We're splitting the chromosomes in half. And this non-disjunction, you get that. All right, so there's your oocyte. On the male side, a similar thing, right? X, Y, you get your X sperm and your Y sperm. If this oocyte gets fertilized by a Y-bearing sperm cell, all that Y chromosome does is make you male. So if you've got the Y chromosome, male, so it's male, but there's one too many X chromosomes. So what we see in Klinefelter syndrome is they're typically sterile. The most notable symptoms are the male reproductive organs are underdeveloped, sort of female body hair pattern, so they don't typically develop like male secondary sex characteristics. Gynecomastia is the development of breast in males. So this is pretty typical of Klinefelter syndrome. Turner syndrome is female, so if we have this oocyte, which didn't get an X chromosome, and it gets fertilized by that X sperm, you have to have at least one X chromosome to be alive. Check. But now there's not another one. So default for the embryo is female anyway. So in Turner syndrome, you only get 45 chromosomes, just the one X. Females with Turner syndrome tend to be shorter, broader chest, lower headline. They always look very young for their age. They typically have sterility, like non-working ovaries. Triple X syndrome. So if that oocyte was fertilized by an X-bearing sperm, you get 3 X chromosomes. There's actually no real symptoms for this. There might be some reproductive issues that occur because, right, you've got one too many in that meiosis thing, but there's no symptoms for that. And hypothetically, because we could get non-disjunction in meiosis 1 and meiosis 2, like hypothetically, you could have up to 5 X chromosomes. I don't think that's common at all, but I mean, you could do that. YO non-disjunction, just 45 chromosomes, only the Y. This is not a viable pregnancy. You have to have at least the X chromosome to make an embryo. Without it, there's nothing. XYY syndrome, this is asymptomatic. They estimate this is somewhere in 1 in 1,000. It's probably less than that. You won't know unless you go have like genetic testing done and you've got one too many Y chromosomes, but it doesn't have any effect on you at all. The other aneuploidy that's considered, again, quote unquote, compatible with life is trisomy 21. That's Down syndrome. And you're familiar with Down syndrome, right? Developmental disabilities, facial features are different. There's some other symptoms with Down syndrome. Down syndrome is, right, in terms of like congenital abnormalities, like congenital heart defects, and then like neural tube issues, and then Down syndrome. All of these aneuploidies are typically associated with the maternal side of this, except for the XYY, that's on the male side. But they're associated with maternal age. The reason for this is meiosis 2 only takes place at fertilization. You've got all of those oocytes by the time you're born. And then puberty, right, and fertilization, you might be older and they've been around that whole time. And the longer that takes that process to occur, the less reliable it becomes. For males, right, it's a conveyor belt. The sperm cells are always being produced. There's always these sperm cells. There's no new oocytes. It's just the old ones getting more mature. So Down syndrome especially is associated with maternal age. Your risk of having a child with Down syndrome goes up as you get older. Other aneuploidies, Tau syndrome and Edwards syndrome, trisomy 13 and 18. They do not have a very good prognosis. There are lots of birth defects that are going to accompany this. That being said, there are children that survive this. There was a guy, for the life of me, I cannot remember his name. He ran for president, probably 2012. I'm not going to swear to that. It's been a while. And he had a kid with, I want to say, Edwards syndrome. And severe deficits like congenital heart defects, excuse me, cleft palate, developmental disabilities, but they are survivable, but prognosis is not great. Most other aneuploidies won't result in a live birth, or they have very little survivability. We don't have time to talk about mosaic aneuploidies. There are things called mosaic aneuploidies where not all your cells have too many chromosomes, just some of them do. We don't have time to get into this. All right, so fertilized oocyte, we got a zygote, and about 36 hours, that thing's gonna start to undergo mitosis. By 72 hours, we have what's called a morula. There's about 16-ish cells in this. And it's like a little ball. And then by day 3 or 4, there's like 100 or so cells. And it reaches the uterus. The blastocyst is what we call this. It's like a sphere, it's like a ball. And there's an outer layer of cells on the outside. And then there's like a little inner cell mass. The outer layer of cells is going to implant in the uterus, and the inner cell mass is going to become the embryo. Oh, good pictures. So here's our zygote, our fertilized egg. Here's dividing. Here's our morula, that's solid ball, little cells. And then here's our blastocyst, and you can see these cells on the outside, and then this inner mass that's going to become the embryo. And here it is about to implant in the uterus.
That's in a week.
That's a week. It's going to float for like 2 or 3 days. Implantation, about a week after ovulation. What does implantation do? Well, it has to adhere to something that it can bind to, right? So the right chemical signals have to be there. Typically, this is going to be in the endometrium. It doesn't have to be. It can be in the uterine tube. It can be outside the uterus. When we break for lab, I've got a MRI pulled up on the table of an ectopic pregnancy. And this ectopic pregnancy, the placenta is attached to the serosa, the outside of the uterus, that outer serosa layer of the uterus. This baby is fairly far along. I would guess that if it's not a viable pregnancy right now, it would be shortly. Obviously, they would have to deliver through C-section, but this looks like it's developing normally. I mean, the fetal skeleton is very clear. Lots of ectopic pregnancies, though, where it attaches outside the uterus, those are life-threatening for both, well, for maternal side. Like, if it's in the tube, like if it's in the uterine tube, the uterus stretches and grows with the developing embryo. The uterine tube does not. So a tubal pregnancy, those have to be removed, or they'll cause maternal death. So we'll look at this ectopic pregnancy, like I said, when we break for lab. Here's our blastocyst. Look at these outer cells, and now they're sticking to the endometrium. Then those cells on the outside, they form two sort of distinct layers. There's a cellular layer that kind of looks like that, and then these cells replicate, and the ones that are on they start to dissolve. This is called the syncytiotrophoblasts. As they dissolve, everything on the inside sort of eats into the wall of the endometrium. So here you see that. So there's our actual cellular layer, and there's all these other cells that are kind of replicating and just dissolving, and as they do, they eat into the wall of the uterus. So, I mean, this endometrium here, you can see like right there, columnar cells. Well, they're gonna respond by just replicating, right? They're gonna regenerate. And so that implanted embryo gets covered by the cells of the endometrium. About 12 days after ovulation, it's fully implanted. And here we see that. So there's our lining of the uterus there, and it's going all the way out. Here we still see that syncytiotrophoblast that's still digesting its way into the endometrial layer. And then here's your actual oocyte or zygote at this point there. Now, remember that in order to keep the endometrium in place, progesterone levels have to stay there. If progesterone levels drop, endometrium's gone. It doesn't matter if that embryo is implanted. If progesterone levels drop, we're done. And the lining of the uterus is lost. So, well, in our cycle there, right, we have follicle stimulating hormone, which causes maturation of the follicle. Follicle releases estrogen. Estrogen causes the release of luteinizing hormone, which causes both ovulation and development of the corpus luteum. And that corpus luteum makes progesterone. And we need that progesterone there to maintain the lining of the uterus. Well, the corpus luteum, without fertilization, is going to degrade and become the corpus albicans. We need a signal that tells the corpus luteum to stick around and to keep making progesterone. That signal is human chorionic gonadotropin, HCG. Those cells of the trophoblast, which will later become something called the chorion, start releasing HCG. HCG acts a lot like luteinizing hormone, and it tells the corpus luteum to stay put, keep making progesterone. We'll see HCG levels continue to increase. They go up sort of exponentially. They should increase daily up until about the second month. And then they'll drop off because now the placenta starts to make its own progesterone and it doesn't need the corpus luteum anymore. So here we see our chart and we see that HCG coming up and it's acting like luteinizing hormone. So progesterone levels are also increasing with the HCG. At this point, the placenta takes over and starts to make its own progesterone. So HCG, this is also the hormone that's detected by your pregnancy test. It's that second line on the pregnancy test. All right, so now the placenta is going to form. The placenta is a combination of both embryonic and maternal tissues. The embryonic tissues, embryonic mesoderm forms this inner cell blast, and they're going to form what we call the chorion and the chorionic villi. The maternal tissues, this functional layer fills these little blood-filled cavities, lacuna, like it's a hollow space. So they're going to fill with blood. The chorionic villi are these little projections that grow from the fetal side out into those blood-filled lacuna. And eventually, the umbilical arteries and veins are going to occupy that space. So they're surrounded by maternal blood. So here we see this. Here's our maternal blood vessels and these big spaces filling with blood. And then here on this side we see those chorionic villi forming, and they're going to be right next to the blood that's pulling here.
So when you have a chorionic hemorrhage, what is that?
That would be here, right?
Just too much blood?
They're breaking.
Oh, okay.
It's breaking off.
So those blood vessels are breaking. So it's a hemorrhage. So it's bleeding into that space. So the endometrium, so that the decidua capsularis is the part of the endometrium that faces the implanted embryo. The placenta is going to be fully formed by about the third month, and it's going to make its own hormones. It's going to make progesterone. It also makes human placental lactogen, which we'll talk about in just a bit, chorionic thyrotropin, and relaxin. And we'll talk about these hormones in just a minute. So here's that. Now, what we see here, here's our chorionic villi, and then here is maternal blood. Here's another picture of this as we develop. Notice our umbilical arteries and veins are coming into the umbilical cord, and then we have all these tiny little capillaries out there. Maternal blood and fetal blood never mix. You're not pumping blood into the fetus or vice versa. We rely on diffusion. What we do with those capillaries is we get fetal blood really close to maternal blood, and we rely on diffusion. So nutrients diffuse in, waste products diffuse out. But the blood supplies never mix. So here again in our chorionic villi.
What happens if it doesn't?
Well, so if you could mix them together, the maternal immune system would respond, right? You would produce antibodies or cytotoxic T cells to fight this developing embryo. And we would be like way back, like there in the cellular level. There are antibodies that cross the placenta, right? That anti-RH antibody crosses the placenta. But that's what we'd worry about, is like that immune response to that. But we've got this barrier here. We've got the membranes of the chorionic villi, then we have the embryonic capillaries. Just like anything else, you've got to go through those cells, and we're diffusing through there. As this happens, the blastocyst starts to convert into what we call a gastrula, and we're going to divide into something called the embryonic disc. There's two layers to this embryonic disc, an epiblast and a hypoblast. And then this sort of turns in on itself and forms the three primary germ layers of the embryo. The epiblast, the upper layer, forms the amniotic sac, the amnion, and it's going to fill with fluid. Amniotic fluid lets the baby float inside. Why? Well, one, it keeps temperature the same, right? That provides a stable temperature. It also prevents the limbs from sticking to themselves as they develop, right? So they don't fuse together during development. Amniotic fluid starts as an exudate from maternal blood. Eventually it's going to be partially fetal urine as the kidneys start to work. From the ventral surface of the embryo, we get something called the yolk sac, and eventually this will become part of the digestive tube. The yolk sac is also the earliest source of our blood cells. The allantois is this little pocketing at the end of this that forms the base of the umbilical cord. And then the chorion we already talked about as part of the placenta. So here is this again, and we can see our layers here. Here is our embryonic disc. There's the chorion being formed on the outside. We've got our epiblast that forms this amniotic cavity up here, and our hypoblast down here that forms this yolk sac. This is 12 days in. 4 days later, you'll notice there's 3 layers of tissue. There's no longer epiblast and hypoblast. We have endoderm, ectoderm, and mesoderm. These diagrams are nice because they kept your color coding the same, like the blue, the red, and the yellow. So you've got the blue, and then the sort of pink, and then the yellow. Gastrulation is the formation of all the stuff on the inside, where we go from being a 2-layered disc to a 3-layered embryo with these 3 germ layers. It starts with something called a primitive streak, and it folds in on itself. This is hard to imagine in 3 dimensions, because what you have here is like this double sphere that kind of looks like that, right? And there's our 2 layers, that becomes the amnion, and that becomes the yolk sac there, and in the middle is that embryonic disc. It kind of looks like that. And here's this thing called the primitive streak. What's happening there is it's folding in on itself and coming back around like that. So now we get 3 layers: ectoderm, mesoderm, and endoderm. So that's that gastrulation. In the middle, there's a thing called a notochord that becomes like a support in the middle of it. It's this rod of cells that looks, and it's not shown very well here. It'll form right here, and you'll see it just like it's a circle of cells. All right, so your 3 primary germ layers: ectoderm, endoderm, mesoderm. These form all the tissues. Ectoderm becomes the epidermis of the skin and the nervous system. Endoderm becomes like the epithelial lining of all the guts. Mesoderm is everything else. So after we form those 3 layers, we're going to start organogenesis. And organogenesis is the formation of all the organ systems. By 8 weeks in, you can see the organ systems. That's the end of the embryonic period. Once the organ systems are starting to form and they're recognizable, it's now a fetus. The first part of organogenesis is neurulation. The nervous system forms. And the way the nervous system forms is the ectoderm, which is, if we look at our 3 layers here, that's this blue layer on the outside. That's ectoderm. The ectoderm is going to sort of fold over. That's not a great picture. Here's our 3 layers again. All right, so here's our ectoderm. So it folds in like this. It like pushes down and folds in. And now it's gonna, those 2 sides are gonna come together and form a tube that we call the neural tube. This starts like immediately with organogenesis. By 22 days in, it's formed a full tube. The top of it's gonna be the brain. The bottom is gonna be the spinal cord.
This is within 22 days of.
Fertilization. So there's our neural tube. Now we talked about neural tube deficits in AMP1, like spina bifida and anencephaly, where that tube doesn't close. So we've already done that, but there's our neural tube forming. Now in the meantime, we have our other cells that are developing. So endoderm, the lining of all the cavities, right? The endothelial lining, it folds in on itself. It's just going to be lining the whole GI tract. Here's another view of this. There's our embryo. Here it is folding in on itself to form the future digestive system, like so. So there in this yolk sac thing here is sort of the primitive guts. There's our neural tube that formed in the meantime. And then here we are at 5 weeks. So mesoderm forms most of the tissues. And the first thing that forms a mesoderm is the notochord. Then we have different cells, somites, intermediate mesoderm, and then lateral plate mesoderm. We're not going to worry about this too much. There are 40 pairs of somites that have these parts, sclerotome, dermatome, myotome. I'm not overly worried about this development. It's just a bit deep for what we've got. Intermediate lateral mesoderm, again, we're not going to worry too much about this. Somata explained it. I'm not worried about the development of mesoderm. What you need to know is big picture. Ectoderm becomes the epidermis and the nervous system. Endoderm, the lining of all the digestive organs and respiratory tract. Mesoderm, everything else. I'm not worried about the divisions of mesoderm like notochords, somites, the intermediate lateral plate mesoderm. We're not going to worry about that. So then we have the fetal development, 9 through 38. Growth happens quickly. So there's an embryo at week 7, about 17 millimeters long. By 8 weeks, 12 weeks, it's more than tripled in size. So there's 8 weeks. 7 weeks, right, 17 millimeters, 8 weeks, about 22. By 12 weeks, 90. And you can see like the sort of proportion starts to even out. 13 to 16 weeks, 140. 17 to 20 weeks, 190. At birth, 360. That's a lot of growth really quickly. So we've got all this stuff going on in the embryo and the fetus. There's a lot of stuff going on the other side as well. Obviously, as the fetus grows, the uterus expands. A question I get a lot is, what happens to all the other guts? Fetus doesn't care. The guts get pushed out of the way. The uterus expands and just shoves everything else out of the way. It's not going anywhere, it's just getting squished. So that's problematic. Because of the weight gain that's happening, and because it's not spread out weight gain, it's all right there, it often causes an abnormal curvature of the spine we call lordosis because your center of gravity's changed. The developing fetus, those tissues are releasing relaxin, right? The placenta releases relaxin. Relaxin is there to relax the ligaments in the pelvis and the pubic symphysis so that those bones can sort of separate out and the baby can go out the birth canal. However, those are not the only tissues relaxin affects. Relaxin is going to affect fibrocartilage and connective tissue in other places, and it makes the joints more unstable. So you get lots of back pain and joint pain because they're not held together as well. You can see here the fundus of the uterus is right up under the xiphoid process. It is pushing up against the diaphragm. Placental hormones. We have placental lactogen, something called chorionic somatotropin. This causes maturation of the breast, glucose sparing in the mother, which means that you spare glucose so the fetus can use that. Chorionic thyrotropin acts like thyroid stimulating hormone, so it increases the amount of thyroid hormone being released on the maternal side and increases your metabolism. Parathyroid hormone levels, vitamin D levels go up so that you bring in more calcium, you're absorbing it, keeping it in the system. The placenta is making enough estrogen and progesterone to make you sick. Sometimes very, very sick. Hyperemesis is when they can't stop vomiting. And then you have the risk of malnutrition and dehydration. Also, it's gross. Nobody wants to be around you when you're throwing up that much. Heartburn and constipation are pretty common because the baby's squishing the stomach and pushing stomach acid back up, and it's squishing the large intestine. It's also squishing the bladder. So you're not just eating for two, right? You're also peeing for two. Urine production goes up because you're also dealing with fetal waste products, and your metabolism's increased. Blood pressure increases. The estrogen from the placenta is going to cause congestion, nasal edema. Difficulty breathing will happen later as the uterus pushes up against the bottom of the diaphragm. Blood volume increases somewhere between 25 and 40%. With that blood volume increase, we get a pressure increase. Venous return might be impaired as well. You've got so much blood, you got a lot more blood, and with that center of gravity change, you can sort of squish veins as well, and it might be harder to get blood back to the heart. So often varicose veins will happen here. Am I selling it? I've often thought about bringing one of my kids up here just to psychologically sterilize everybody.
Yeah.
I just I don't know, the little guy's pretty cute. I've got one of them he's a handful, but the rest of them are pretty mellow. So birth, parturition, labor, whatever. So the last few weeks of pregnancy, the fetus is making cortisol. Cortisol tells the placenta to make more estrogen. That estrogen is going to trigger the production of oxytocin receptors in the muscle of the uterus. Oxytocin, if you'll remember, oxytocin comes from the posterior pituitary. It binds to those receptors and it causes the muscle to contract. This causes what we call Braxton Hicks contractions. They're small, short contractions of the uterus. Well, this starts off a cycle because that oxytocin is making the uterus contract. That's uncomfortable. And the stress from that causes the release of oxytocin, which makes the uterus contract, which is uncomfortable. And it's a positive feedback cycle. Not only that, we're releasing surfactant. The baby's making surfactant at this point, like those last 2 weeks, making surfactant. The surfactant is going to allow the surface tension inside the lungs to be reduced so the baby can breathe, but it's also going to soften the cervix, and the cervix is going to start to dilate. Fetal oxytocin is going to be produced. Fetal oxytocin causes the placenta to make prostaglandins, which also make the uterus contract. So now, with that uterine contraction, the more uncomfortable it is, the more oxytocin is released, which makes the next contraction a little more vigorous, which causes the release of oxytocin, which makes the next contraction, and it's positive feedback. So here's that. So oxytocin, stimulation, positive feedback. The cervix dilates. It's fully dilated at 10 centimeters. And during this time, the contractions are starting to get more vigorous and closer together. Initially, it's really weak, but positive feedback cycles amplify very, very quickly. So, cervix dilates, the amniotic sac will rupture. Right, the water breaks, amniotic fluid, and then the head enters the pelvis. And here you see the head entering the true pelvis there. Later in dilation. Contractions happen about every 2 to 3 minutes, they're about a minute long, and then you're gonna push the kid out. Congratulations, it's a boy. But it's always a boy.
I feel like they did the Yankee Night.
Will they always come out face down?
Most of the time, they try to manipulate it if not, but I mean, it doesn't have to. And like, you know, there are breech births as well where they come out feet first.
My daughter was a breech. They had a mess with her to flip it around.
Yeah, they try to manipulate it so you flip it around, because this is the optimal position here. It's much more difficult when it's a breech birth.
Do their shoulders, like, how do they fit through? Do they like
So-
dislocate?
No, the shoulders aren't like dislocating. That you,
It's
if
like real.
that's a possibility, those shoulders can get stuck, and that's a serious complication there that might. This sounds awful. That might involve pushing them back in and surgically. Yeah.
Do not. Oh my gosh.
But- Right, because if those shoulders get stuck, like it's very dangerous for both the baby and the mother. So but hopefully relaxing has relaxed this enough so that pubic symphysis separates out enough where this can happen.
So like in this little picture, like the shoulders kind of low.
Or like they're like this or something.
Yeah, we're not even gonna, like, there's some incisions that can be made here to aid that along. Yeah, that episiotomy, if you're not familiar with that, don't Google that.
Mine was 4 steps down,
Oh my god.
literally. And she tried to hide them coming into that room, like by her
The
sides.
process-
And I was like, I can
Because
see
you
those.
pick out.
They are.
It's like it's,
Yes, it's awful.
You're like, those are gonna fit in me?
I mean, there's a kid in the right house.
They make it work.
And his head was like this.
Did you get
All of my kids have been C-sections. So like I have.
They were all C-sections?
All of them. So I have no real experience. I'm never like this miracle of life crap, like no, like C-section. Knife goes in, baby comes out. It's magic. So
They pull like this, like her intestines out, right?
well they just not like
like
not
shoulders.
like
I
guts.
know, but like,
No, they're not outside.
No,
I've
not
seen a
outside.
video where they've like taken it.
Well,
Like,
okay, so they move stuff to the side. That's like omentum and stuff they're moving to the side. The
Oh.
uterus takes up most of that space. Like it takes up all that space. So it's gonna be like first and foremost. Like you don't have to really move intestines out. The intestines are not in front of the uterus. Like if you go back and look, like the uterus is in front of everything, like there.
Yeah.
Like, all the guts are shoved back. So that's like what's right there.
Okay.
There are things that
Thing is your placenta.
Yeah.
so that's what they're taking out?
yeah,
Oh, okay,
yeah.
okay. So they
You
put
can feel that come out.
Oh my gosh, when you have to push down.
So that placenta has to come out afterwards, right? That positive feedback cycle continues even after delivery, so that the placenta comes out. So oxytocin is still being released, and uterus keeps contracting, and then the placenta is delivered after the kid. And if it's not, bleeding will continue. So there's the placenta coming loose.
Oh, yeah.
How does sperm get stuck in you? Yeah.
As it gets stuck.
Oh, okay.
So it shouldn't, but I mean, there are fragments that could, but
Yeah.
like, if that happens, like, they've gotta make sure that it's all out.
It was a big relief, honestly.
Them pushing on your stomach has to be the worst thing.
Oh, yeah, especially after life.
I have a picture of them pushing on my stomach, like, I guess my friends were taking a picture of me, and they were pushing on my stomach, and my face was like, wanting to kill me.
Right, so they're trying to push, like, get the uterus to contract back. And that contraction of the uterus is gonna continue even after the kid's born. And that's still, there's still going to be oxytocin that's being released. And we'll look at this in just a second, but oxytocin is released as part of the milk letdown reflex. And that reflex also causes the uterus to continue to contract back to its original size. So right, the kid's nursing, and those contractions are still happening, and the uterus is contracting back to its original shape there. So for the baby, like right after there, you have the neonatal period. They do an APGAR score, like they give your kid a score. I guess they rank him on a scale of 1 to right, 1 to 10. So then you know like where you rate there. The kid's got to breathe, and we've already talked about this. So carbon dioxide is your major respiratory stimulant. Kid's not breathing, cut the cord, so we're not getting any diffusion of oxygen across anymore. Carbon dioxide immediately starts to build up. We get acidosis, respiratory center fires, and they have to breathe. And so then it's not an easy thing for them, but hopefully we're far enough along that there's surfactant being produced, surface tension's reduced, and then they breathe. And they'll breathe pretty fast for the first couple of weeks. 6 to 8 hours after birth is called the transitional period. It's pretty unstable. They're asleep, they're awake. Vital signs are all over the place, like body temperature, blood sugar. There's all sorts of things that are all over the place. And then it pretty much stabilizes after that. We've already talked about these fetal blood vessels. The umbilical arteries and veins become constricted, they fibrose. Some of them will stay, like the ones inside. So the proximal arteries become arteries that go to the bladder. The distal arteries end up becoming umbilical ligaments, which are not really ligaments, it's just scar tissue. The ductus venosus becomes the ligamentum venosum, that's the bypass for the liver. The foramen ovale becomes the fossa ovalis. We talked about this with the cardiovascular thing. The umbilical vein on the inside becomes the round ligament of the liver, which we looked at in the liver. So that ligament that sort of hangs down under the liver is a round ligament. And that's used to be the umbilical vein.
Like actual benefits of delayed cord clamping or whatever?
Like when they leave it?
Yeah, well, not like leave it for a long time, but just they don't cut the umbilical cord for like a little bit, so it gets like soggy.
I have no idea if there's any benefit to that or not. I have seen things where people like leave like they leave the cord and the placenta, like
yeah.
Which
That would cause infection, wouldn't it? 'Cause it's like
I don't know how that works actually. Like I saw this whole thing where they were carrying around like the baby, and then with it like they had like the placenta like I don't know, wrapped in saran wrap or something, and so they just carried it around. I don't know if there's any benefit to that. I wouldn't think so.
Yeah, they're just good for that stuff. Okay, I'm like, I did it. I didn't know what I was like, if it's their blood, let them have it. No, I don't know. I didn't know.
Oh, like are we talking about banking the cord blood?
No, I know about banking the cord blood because of the stem cells.
Right.
But I just heard that there was like a benefit to delayed cord clamping to let the blood like actually go into the baby before you cut it off.
Ew.
I don't know.
Chunk that.
Oh, that one is trash.
Yeah, people do eat theirs too. Or put it in capsules. Put it in capsules.
Yeah, that's a thing.
Is there any benefit to that?
Oh yeah, is there really a benefit to that?
Not that I'm aware of. I mean,
I
so
thought it was weird.
the thought process there is we see this in the animal kingdom, like they deliver the placenta, and then like quite often they'll eat the placenta. Good news, we're people, so
Natural reaction.
you don't have to, right? For that, the hypothesis there is that right there's energy expenditure that's gone into making that, and so for the maternal side, it's going to be difficult. Right, they just delivered a giraffe or whatever, and they're taking care of the infant animal, and so the resources for them are limited, and that's a resource right there. Somebody's gonna bring you food.
Yeah.
Right, so we should be fine. There's probably not so much of a benefit to that. And like encapsulating it and drying it, I don't think there's really any sort of
They pay some good money for that.
research that's behind that, but I mean,
I thought it was weird, but.
if you can keep it and sell it, rock on.
So,
There's some weirdos out there.
I got some pills for you.
It's probably illegal as everything. I'm not supporting this by any means.
Black market.
All right, so lactation, we've got prolactin from the anterior pituitary that's caused by the hormones for the placenta. So the estrogen, the progesterone, placental lactogen causes the hypothalamus to release its hormone. That's a releasing factor. Then the anterior pituitary releases prolactin. So prolactin causes milk production. Now the first milk that's produced is called colostrum. It's kind of a yellow fluid. It's thicker than the true milk. It's got lots of vitamin A in it. It's got lots of antibodies. Antibodies are going to be conveyed through breast milk. Remember, that's passive humoral immunity. That's immunoglobulin A. Passive humoral immunity. Positive feedback again. Oxytocin's released. Oxytocin causes the milk letdown reflex. It also causes uterine contraction, but it's positive feedback. The more the infant nurses, the more of that lactation and letdown that you're going to get. So there's your positive feedback. In terms of like nutrition, breast milk is the easiest absorbed form of nutrition, as opposed to like milk from other animals. It's in a form that's easier for the infant to digest. Also, we have passive humoral immunity, immunoglobulin A, those complement proteins, interferons even that are going to cross over and provide some immunity to the newborn. Also, interleukins, prostaglandins help reduce inflammation. It helps eliminate what we call meconium. When you have a kid, like the first time it poops, it's like black tar. That's meconium. And breast milk helps eliminate that and to prevent jaundice. And it encourages colonization for the large bowel. All right, so now with this, we need to talk about one more thing before we split. This one is, we're gonna touch on this, and I'll go ahead and upload these notes for your Blackboard. But we're just gonna talk about it just to wrap things up. We're gonna skip the middle, right? All the growing up and puberty crap, we're skipping this. Go straight to getting old. So I like this because I need to tell you guys, you're gonna get old, it's gonna suck. When I started teaching, I was like 27, and I was like, no. And now I'm like, oh, yeah, it's gonna suck, guys. Like, you're like, it's not gonna be me. Yeah, it is. Yeah, it's totally you. Currently, like 1 in 9 Americans is older than 65. The thing with being old is that 80 to 90% of your healthcare costs that you will incur in your lifetime happens during the last 2 years you're alive. So we have an entire branch of medicine dedicated to just old people. Geriatrics. I know lots of people in geriatrics. It's a rough job because all your patients are, getting old and dying. But it's a specialized branch of medicine. Taking care of people as they age, because we do have all these conditions that occur with the senescence of systems. So we measure age in years, right? I think this is still the case. The oldest person recognized by like the Guinness people is like 122. In the U.S., and this is pre-COVID. I don't actually know our stats right now, but pre-COVID, the average lifespan in the United States was around 78. Women tend to live longer than men. On average, about 5 years longer than men. Here's a map. This, I don't remember what year this is, but this was life expectancy. Again, pre-COVID. COVID's thrown us as a loop, but pre-COVID, the United States was 49th on the list for life expectancy. 48 countries where people live longer than we do. The highest average life expectancy is Monaco. Average life expectancy is about 90, on average. What are they doing in Monaco? What do you know about Monaco?
They're super rich.
They're freaking rich. It is a ridiculously wealthy place. Like, I'm pretty sure most of it's also a really tiny country. Like, most of it's occupied by like giant casinos. Like, it is where the rich people go and live on their yachts or whatever, and drive Ferraris around. So yeah, crazy crazy there. But countries that are better than us, Canada, like most of Europe, Japan, Australia. The lowest on the list was Angola, sub-Saharan Africa, where the average life expectancy was about 38.5 years.
That's crazy.
So you got other things, like you've got like instability in terms of like governmental instability and war, and you've got the HIV epidemic. So why do you get old? Like what's the purpose? Like why do we die? You don't have to, actually. Like in terms of being alive, like as people, yes, you have to die, sorry. But things that are alive don't have to die. How long do trees live? I don't know. It's a long time. There's a species of jellyfish that after it mates, it reverts back to an immature form, and hypothetically, it's immortal. I mean, something's gonna eat it and kill it probably, but barring that, it would live forever. Like, yeah, things can live a lot longer than you would expect. Or, you know, again, trees might live forever too, as far as we know, like some of those giant trees. So, what's the purpose? Like, why are we dying? It has to have a purpose.
Sales.
What would happen if we didn't die? Overpopulation, right? There would be too many of us. So by dying, like we're allowing other generations to continue. And it is built into our genetics. Senescence is the degeneration of the organ systems that happen over time. And it is pre-programmed into your genes because every time that DNA replicates, it slices off the end of the strand, that telomere that's at the end. Immortal cells like stem cells and things, there's an enzyme there called telomerase that puts it back. When we do this in the lab, like we make immortal cell lines by adding that gene back or reactivating that gene. But for other cells, as they replicate, they get shorter. And eventually, they can't do that anymore because they've cut away all those telomeres. So at the cellular level, aging's how many times that cell divides? We've done studies with mice where we do caloric restriction, and of course it stunts the growth of the mouse, and they're malnourished. But right, they're just getting enough nutrition to stay alive. You can increase their lifespan by about 50%. Now they're super tiny, and it's gotta suck to be them because they're super hungry. But I mean, yay, they get to be an older mouse. This is not something that we're gonna pull off. Like as people, we're not gonna be like, oh, don't feed that baby. They're gonna live to be 150. So organ systems age at different rates. Most of them reach their sort of peak function 20s, 30s. And then after that, stuff starts to slow down. One of the slower systems nicely is the nervous system. From 30 to 80, nerve transmission speed goes down 10, 15%. Number of nephrons working in the kidney, they drop by about 60%. One of the more prominent features that we see in people that get older is senescence of the skin. Like 2/3 of people older than 50 and pretty much everybody older than 70 has a medical complaint about the skin. You'll start to notice senescence of the integument around 40, late 40s. The hair gets thinner, it turns gray as melanocytes die. The skin gets drier, the hair gets drier, those sebaceous glands atrophy. Replication of the epidermis slows down, which means that healing for the epidermis slows down. It also means the epidermis gets more fragile. And you'll see this in old people. The skin looks much thinner. It's nearly translucent. It's kind of shiny. And like you can see, it looks like wax paper. In addition to that, the blood vessels there are more fragile. And you'll see this in old people, like they'll bump up against something, giant bruise, like where those blood vessels under the skin break. It's very easy to like open wounds, and it takes forever for them to heal. This is all pretty much normal. What's gonna happen? This is intrinsic aging. It happens with time. We can slow it down. obviously, like there are various treatments available to slow down the aging process of the skin. If you watch the football game with Dolly Parton, there are treatments available to stop aging or slow down aging of the skin.
So she didn't got the problem.
Right, so there's like that, or like you see like this is probably a dated reference for you, like Cindy Crawford, like I don't think she's changed in like the last 30 years.
She drinks some blood.
Right, so there's like a painting somewhere in a closet that's getting old, like.
Yeah.
So yeah, and you see this like where people, especially like that, where there's obviously some treatments available there, and they take really good care of their skin. But not taking care of your skin is going to amplify this process greatly. Photo aging is when you're exposed to UV light. 90% of that senescence is coming from that UV exposure, right? It will age you. That exposure to the sun and that ultraviolet light amplifies the wrinkling of the skin and how dry that is. So yeah, take care of your skin. The bones degrade because the osteoblasts slow down. This is amplified in women because of menopause. We have osteoporosis. After 40, women lose about 8% of their bone mass, men only about 3%. And then of course, like osteoarthritis, about 85% of people that are older than 75 have some degree of osteoarthritis. The muscles atrophy with age, and muscle starts to get replaced with fat. This is something that again is inevitable. It doesn't matter what you're doing. I guess it's gonna have to be, you're gonna keep working out forever. Great. So when you think about that, working out forever, I want you to look at pictures of like Ric Flair now versus Ric Flair in 1994. And like you'll see like videos like Ric Flair still like lifting weights and working out, awesome, but he's a lot different now than he was because those muscles have begun to atrophy over time, and there's nothing that you're going to do to stop that. You can slow it down definitely, but eventually you're going to have that, and like you're not going to have the performance when you're 70 like you did when you were 30. So muscle mass and strength peaks in the mid to late 20s and degenerates as the muscle fibers age. Those sarcomeres aren't so organized, and the myofibrils start to go down. Mitochondria get smaller. They're not as good at doing their job anymore. The nervous system, we have senescence of the nervous system. Fewer motor neurons also contributes to that muscle atrophy. Your nervous system peaks in your mid-30s. Your brain's actually still developing in your 20s, and in the 30s you kind of hit a high point, and then things start to slow down. Now, on average, the brain's like half the size at 75 as it was at 30, but that's including like pathophysiology. So, and that would include Alzheimer's disease. And in Alzheimer's disease, we get significant atrophy of the brain. So we talked about Alzheimer's in AMP1, so I'm not going to super rehash this a lot, but we get the accumulation of this peptide in the brain. Here we see that, those little tau particles there as the microtubules disintegrate, and we lose synaptic plasticity. You're familiar with Alzheimer's, the ability to form new memories or to concentrate, and then eventually the old memories degrade, and eventually other functions of the brain start to slip off. Eventually Alzheimer's is terminal, but a lot of times death occurs from like other things like infection or like respiratory tract infection, something like this. But eventually, even without that, Alzheimer's would be fatal. Here's a thermal scan of the brain with Alzheimer's, and you can see like the sort of areas that are still active and then those that are not. Here's a cross section. You can see the enlargement of the ventricles, shrinkage of the cortex. Here's actual. So these people have been the same age. Alzheimer's brain, normal brain.
They're the same age.
Yeah. So there's a giant loss in function here as we get this atrophy.
It's just like crippling pretty much?
Yes,
Yeah.
eventually it is. The Alzheimer's disease is like the leading reason people are placed in the long-term care. And it's difficult. As you watch that degradation of function, and it slowly sort of slips away. They become less and less independent. They become a fall risk. There's just lots of complications from this. All the other systems will degenerate somewhat with age. Like I said, the kidneys a lot. The immune system, we talked about this when we were talking about immunity. Like we have autoimmune diseases with age, and immunodeficiencies as well. The circulatory system, right, the whole cardiovascular system, we have all the aging that's going to happen there. Atherosclerosis, obviously your risk goes up with age there, and hardening of the arteries. Endocrine system, especially for females after menopause, the endocrine function's kind of thrown off without those hormones. Everything's going to get older. So, yeah, I like to end on a high note now. So we'll come back on Tuesday and we'll talk about infections, and then on Thursday we'll review.